There are two maps in front of you. That is where this story begins. Not with a discovery, not with a headline, but with two images placed side by side in a research laboratory sometime in the early years of this century by a scientist who would later say that when she first saw them together, she felt a cold and inexplicable unease she could not immediately name. The first map traces the movement of human populations across the face of the earth over the past 200,000 years. It is built not from footprints or road signs, but from something far more durable, the quiet, patient record encoded inside the human genome. Every cluster on this map represents a population. Every line connecting those clusters is a migratory corridor, reconstructed from the shared mutations that living people carry without knowing they carry them. Inherited from ancestors who walked paths that have long since been swallowed by desert, ice, or rising sea. The second map is entirely different in origin. It is a visualization of the internal state of a deep neural network, a computational system trained on millions of data points to recognize and respond to complex patterns. The clusters on this map are not people. They are artificial neurons, mathematical abstractions connected by weighted pathways that shift and strengthen and fade as the system learns. The lines between them trace the movement of information from input to output, from raw noise to refined meaning. Neither map was created with the other in mind. The first was assembled by population geneticists over decades of painstaking fieldwork, laboratory analysis, and careful statistical inference. The second was generated by a machine that had no knowledge of human history, no awareness of continents or centuries, no concept of survival or migration or loss. And yet, when you place them side by side, the shapes are identical, not similar, not loosely comparable. Identical in the mathematical sense that two objects can be identical when the equations describing them are the same, when the measurements you take from one could have been taken from the other. At the center of each map sits a dense, highly connected cluster of nodes, rich with pathways and thick with traffic. Radiating outward from that center are progressively sparser regions, populated by smaller clusters that are connected to each other, but only weakly connected to the core. The information flow, whether it is genetic or digital, biological or computational, moves from the center outward, transforming at each step, arriving at the outer edges of the network in a form that barely resembles what entered at the beginning. There is no satisfying explanation for why this should be true. You could spend an afternoon with the world's most distinguished computational biologists and population geneticists, asking each of them to account for the resemblance, and you would receive responses ranging from the intriguing to the speculative to the quietly, professionally unnerved. Some would tell you it reflects universal principles of network formation, that any system moving information across space tends over time toward these same structural arrangements. Some would tell you it is a mathematical inevitability, the product of shared constraints operating on very different substrates. Some, the most honest ones, would tell you simply that they do not know, and that not knowing keeps them working. What you are about to hear is an investigation into that resemblance. It will not stay on the surface. It will not be satisfied with the observation that two maps look alike and leave the question there. It will follow the parallel down through every layer, through the mechanics of genetic mutation, through the mathematics of ancient movement, through the deep structure of how neural networks learn, until the resemblance either dissolves into coincidence or resolves into something far stranger and more significant. Before the first word of explanation, though, the question deserves to hang in the air for just a moment longer. Two maps, one drawn in the slow language of heredity across 100,000 generations of human life. The other rendered in milliseconds by a machine built from silicon and electricity and the accumulated knowledge of our species. And yet both of them somehow show you the same thing. Why? To understand the resemblance. It helps to understand what each system actually is, not in technical language, but in the language of what it does, what it is trying to accomplish, and what constraints it operates under. The first system is ancient human migration. It is, in the most literal sense, the longest story ever told. Over the course of roughly 200,000 years, beginning in sub-Saharan Africa and ending, if it has ended, in every corner of the habitable earth, human populations moved. They moved because climates shifted and familiar environments became unlivable. They moved because prey animals led them into new territory. They moved because younger generations drifted slightly further from the places their parents had been. And over enough generations, that drift became displacement, and displacement became colonization. No individual making any of these small movements knew they were participating in an epic. The epic only becomes visible from a great distance, looking backward across geological time at the accumulated trace of millions of ordinary lives. What this migration left behind, its primary record, more complete than any cave painting or fossilized bone, is the human genome. Every person alive today carries, embedded in their DNA, the ghost of every route their ancestors walked, every population they belonged to, every encounter they survived. The genome is a logbook of the journey, written in a language that only became readable in the last few decades, and what it contains has repeatedly overturned everything historians and archaeologists thought they knew about where we came from. The second system is the artificial neural network. It arrived in the 20th century as an idea before it arrived as a machine. The idea that intelligence might be less like a calculator and more like a brain, less about following rules and more about recognizing patterns built up through experience. The artificial version of this idea is architecturally simple. Layers of connected nodes, each one performing a small calculation passing its result to the next layer. With the strength of each connection, its weight, adjusting gradually in response to errors until the whole system becomes capable of things that no individual part could accomplish alone. These two systems appear, on the surface, to have nothing to do with each other. One is biological, the other is computational. One unfolded across hundreds of centuries, the other trains in hours. One is written in the chemistry of living cells. The other runs on electricity through refined sand. The comparison seems, at first, like the kind of loose analogy that scientists reach for when they want to make an abstract concept feel more human. A metaphor, in other words, not a measurement. But here is the thing about the two maps. A metaphor produces a resemblance of appearance. It makes one thing look like another thing. What the two maps show is not a resemblance of appearance. It is a correspondence of structure. The same graph theoretic measures, the same mathematical quantities that describe how information flows through a complex network, return the same values when applied to both systems. This is not a poet observing that two rivers look alike. This is a mathematician demonstrating that two rivers have identical flow equations. Population clusters in the migration record correspond quantitatively to hub nodes in trained neural networks. Genetic bottlenecks correspond to dropout events. Gene flow between isolated populations corresponds to backpropagation across layers. These are not impressionistic parallels. They are structural identities, each one accompanied by a number and a formula and a published paper with reviewers who pushed back and ultimately could not find the error. Something is happening here that is genuinely difficult to explain. The story ahead will try to explain it. But first, it is worth pausing to appreciate the full strangeness of the situation. Because what becomes ordinary as an explanation was, at first encounter, profoundly and productively strange. Two systems, one pattern, and a question that neither field has fully answered. Is this convergence a mathematical curiosity, or is it telling us something fundamental about the nature of intelligence itself? Consider the eye, the vertebrate eye, the kind of eye carried by fish and frogs and falcons and human beings, evolved once in a common ancestor several 100 million years ago. The octopus eye, however, evolved independently. A separate evolutionary line, starting from scratch, working with different genetic material over a completely distinct history, arrived at almost the same solution. Lens in front, photoreceptors at the back, a system for focusing light and translating it into signal. The physics of light demanded the architecture. No matter where you start or what materials you use, if you are trying to build something that turns light into information, you will end up with something that looks very much like an eye. Biologists call this convergent evolution. The same environment, the same physical problem, the same set of possible solutions, and independently evolving systems, finding the same answer without any communication between them. It happens everywhere in nature. Wings, echolocation, venom. Transparency in deep-sea creatures. The universe, it seems, has preferences. When it encounters a particular class of problem, it tends to solve it in a particular class of way. The question this documentary raises, and pursues with full seriousness, and does not pretend to answer easily, is whether something similar might be happening at the level of information processing itself. Whether the problem of moving information efficiently through space and time, whether the challenge of encoding what has been learned so that it can be retrieved and used, whether the need to build a system that is simultaneously adaptable and robust, whether all of these problems, like the problem of capturing light, might have only a small number of deep solutions, and whether both biological evolution and artificial intelligence, arriving at these problems independently, might be finding the same ones. This is not a claim about design. It is not a claim that the universe is directed toward any particular outcome, or that human history was orchestrated toward any goal. It is a narrower and more precise claim that the mathematics of efficient information transfer may be universal in the same way that the physics of optics is universal, and that two systems, one biological, one computational, that have both faced the problem of distributing complex information across a network in a noisy, unpredictable environment, may have both converged on the same structural architecture for the same reason that two unrelated species converged on the same eye, not because they copied each other, because the problem left them no better option. What follows is an investigation into that possibility. It will move slowly and carefully because the claim is large and the evidence needs space to breathe. Chapter by chapter, it will build a picture. First, of what the human genome actually records and how scientists read it. Then of the physical routes that ancient humans traveled and the patterns those routes left behind. Then of how neural networks are built and how they learn. And then, finally, of the specific and documented correspondences between these two systems that have led a growing number of researchers to believe that they are not merely analogous, but mathematically equivalent. Along the way, there will be surprises. There will be moments where the evidence turns in an unexpected direction and the story has to follow. There will be data points that, when stated plainly, are difficult to believe, not because they are speculative, but because the implications of accepting them are so large that the mind instinctively resists. Those are the moments worth staying for. The question is on the table. The investigation begins now. Imagine a letter, a very long letter, 3 billion characters long, written in an alphabet of only 4 symbols. Now imagine that this letter is copied by hand, generation after generation, by scribes who are careful but not perfect, so that with each copy, a small number of characters are transcribed incorrectly. most of the errors are harmless. They fall in stretches of the letter that do not change its meaning. Some of them alter a word in a way that makes the sentence slightly different, though still intelligible. A very few of them change something fundamental. And because the scribes pass their copies on to their children, and the children's children copy those, every version of the letter that exists today carries a unique record of all the copying errors that accumulated on its particular line of descent. This is, in essence, what the human genome is. The 3 billion base pairs of DNA that sit inside the nucleus of every one of your cells constitute the longest text ever written, and the most carefully preserved, and the most informative, and the most misunderstood. For most of human history, the text was unreadable. It took until the end of the 20th century to develop tools capable of sequencing it at all, and well into the 21st century before sequencing became cheap and fast enough to read it in large populations. When scientists finally began reading the letter in earnest, they discovered that what it contained was not only instructions for building a body, it was also encoded in the pattern of accumulated copying errors, a complete travel diary of every ancestral line that had ever contributed to that body. Every copying error, the molecular biologists call them single nucleotide polymorphisms, though you need not remember that term, is a timestamp. When a new variant arises in a population, it first appears in a single individual. If that individual survives and reproduces, the variant spreads to their descendants. If the population later divides, one group staying, another moving, the variant goes with whichever group carries the descendant. Over generations and centuries, the distribution of that variant across living populations tells you something about where the original individual lived, how the population split, and which groups ended up sharing a common ancestor. This is not guesswork. It is probability applied to biology, and it is extraordinarily precise. When geneticists compare the genome sequences of people from different populations around the world, they can calculate with remarkable accuracy not just whether two populations are related, but when they diverged, how many people were involved in the separation, and whether any subsequent mixing occurred. The human genome is not just a record of who a person is. It is a record of every place their lineage has ever been. The most striking demonstration of this is also the simplest. There is a particular genetic variant, one specific letter change in one specific position in the genome, that is found in virtually every human being alive today whose ancestry lies outside of Africa and almost nowhere else. It is not found in Central African populations or in populations whose lineages never left the African continent. It appears at high frequency in people whose ancestors crossed out of Africa into the broader world, and it is absent in those whose ancestors did not make that crossing. Scientists have dated this variant to approximately 60 to 70,000 years ago. Somewhere in East Africa, in a population that was about to begin the longest journey in human history, a single copying error occurred. It spread through that population. And when a group from that population eventually crossed into the Arabian Peninsula and became the ancestors of everyone now living outside Africa, they carried it with them. Every generation since then has passed it forward, faithfully, through every empire and every plague and every upheaval that human history has contained. You carry it now, if your ancestry comes from anywhere outside the African continent. It is one letter in 3 billion, and it is the opening line of a story that this chapter will now begin to tell in full. Individual copying errors are useful, but they become far more powerful when they are grouped. Over time, scientists discovered that certain clusters of genetic variants are almost always inherited together, passed down as a unit, generation after generation, along specific ancestral lines. These clusters are called haplogroups, and understanding them is essential to understanding how the human genome functions as a migration map. Think of a haplogroup as a compressed data packet. It contains within it not a single piece of ancestral information, but a whole bundle, a record of dozens or hundreds of related variants that arose in a specific ancestral population at a specific time and have been traveling together through descendant lineages ever since. When a scientist identifies your haplogroup, they are not just identifying one letter in your genome. They are identifying the ancestral population you belong to, the broad route your lineage took, and the approximate period during which your ancestors were living and moving in a particular region of the world. The haplogroup system works differently for maternal and paternal ancestry. Mitochondrial DNA, a small, separate set of genetic material that sits outside the main genome and is passed down exclusively through the maternal line, accumulates its own distinct set of variants, giving scientists a direct window into the ancestry of mothers and grandmothers and great-grandmothers stretching back thousands of generations. The Y chromosome, carried only by men and passed exclusively through the paternal line, provides the equivalent window for fathers and their fathers. Together, these two systems allow researchers to build two independent trees of human ancestry, one maternal, one paternal, and compare them for consistency and contradiction. What emerges from these trees is a picture both breathtaking and structurally strange. All living human mitochondrial lineages trace back to a single woman who lived in Africa, somewhere between 100,000 and 200,000 years ago. She is not the only woman who was alive at that time, not by a long way. She is simply the one whose direct maternal line has survived unbroken to the present. Every other female lineage from that period has been extinguished somewhere along the way, leaving no living female descendants. The fact that her line survived is not evidence of anything special about her. It is the expected outcome of the mathematics of lineage survival in a finite population. But the fact of it is still quietly astonishing. The haplogroup trees do not grow evenly. Some branches are enormously thick, encompassing hundreds of millions of descendants. Others are slender threads, maintained by small isolated populations that have barely grown since the initial split. The distribution of branch thickness across the tree is not random. The thickest branches correspond to the populations that, at some critical moment in history, occupied a geographic position or survived a demographic event that allowed them to expand dramatically, to become, in network terms, hub nodes. The thin branches correspond to populations that remained isolated, that passed through severe restrictions, or that simply found themselves on the wrong side of a geographic barrier when the conditions for expansion arrived. Among the most significant of these branching points is a haplogroup called L3. It sits near the base of the human mitochondrial tree, not at the root, but close to it, and it represents the common ancestral lineage of every human being who has ever lived outside of Africa. Every European, every Asian, every person of the Americas or Oceania, regardless of how different their cultures, their languages, their appearances, shares descent through L3 from a woman who lived in East Africa between 60 and 80,000 years ago. Scientists can date the emergence of L3 with reasonable confidence because mutations accumulate at a roughly constant rate, providing a kind of molecular clock. They can locate its geographic origin because its closest relatives are still found predominantly in East African populations, and they can trace its subsequent diversification into the dozens of haplogroups that now populate the rest of the world, each one marking a specific direction of travel, a specific period of movement, a specific population that diverged from the mainstream. The haplogroup tree is, in this sense, the most complete historical record in existence. It predates writing by 50,000 years. It covers every human population on Earth, and it is updated continuously, automatically, by the biology of reproduction itself. Every new birth adding one more data point to a record that began before the first human foot ever left the African continent. For decades after population genetics established its basic framework, the field operated almost entirely on living subjects. Scientists would collect blood or saliva samples from people in different populations, sequence the relevant portions of their genomes, and build trees from the resulting data. The method was powerful, but it had a fundamental limitation. It could only see the past through the filter of the present. If a population had been completely destroyed by disease, by conquest, by simple attrition, it left no living descendants to sample, and its genetic contribution to the broader human story was invisible. Then came ancient DNA. The extraction of genetic material from archaeological specimens, from bones and teeth that had been lying in the ground for thousands of years, began as a technical curiosity and became, in the span of 2 decades, the most disruptive force in the history of human evolutionary science. The first successful ancient DNA extractions were crude and prone to contamination. Modern techniques, developed largely in the past 15 years, are precise enough to sequence entire ancient genomes with a reliability comparable to modern clinical sequencing. The result has been something close to a revolution. What the ancient DNA record contains is, in many ways, more complex and more disturbing than the clean narrative that living DNA research had constructed. The story it tells is not one of populations moving in orderly succession across a fixed map. It is a story of collisions, replacements, disappearances, and resurfaces, of populations that dominated a region for thousands of years and then vanished from the genetic record almost overnight, replaced by incoming groups whose origins lay thousands of miles away. Ancient Britain, ancient Europe, ancient South Asia. Everywhere the ancient genome record has been examined with sufficient resolution, what emerges is not a palimpsest of gradual accumulation, but a series of sharp discontinuities, each one representing a moment when the old population was largely displaced and a new one took its place. But the deepest disruption came not from the complexity of human movement, but from a finding that most geneticists initially dismissed as contamination. Scattered through the genomes of modern humans, particularly in populations outside of Africa, are small but consistent segments of DNA that do not match the expected modern human pattern. They match something older, something that should not, by the conventional account of human evolution, be there at all. They are Neanderthal, and in some East Asian and Pacific Islander populations, they are also Denisovan, genetic material from a separate archaic species known only from a finger bone and a few teeth found in a cave in Siberia. Modern humans interbred with Neanderthals. They also interbred with Denisovans. The evidence is now unambiguous, replicated across dozens of independent studies, visible in the genomes of billions of living people. In populations of European and Asian descent, roughly 2% of the genome carries Neanderthal origin. In some oceanic populations, the Denisovan contribution reaches 5% or higher. These are not marginal signals. They are substantial functional contributions to the modern human genome, including variants that affect immune function, fat metabolism, and adaptation to high altitude. The implications for the migration map are profound. The story was never a single, branching tree of pure Homo sapiens lineages moving outward from Africa in clean succession. It was a network, a complex, tangled web of meetings and partings, of genetic material flowing between populations that was separated by tens of thousands of years, of independent evolution and then thrown together at the edges of expanding ranges. A network is a fundamentally different mathematical object from a tree. Trees branch but never rejoin. Networks branch and rejoin and cross and loop back. The mathematical tools needed to describe a network are different from those needed to describe a tree, and as it happens, they are very similar to the tools used to describe the internal architecture of a deep neural network. That observation, quiet as it is, will become important. 70,000 years ago, the world looked different. The Arabian Peninsula, now largely arid, was periodically laced with river systems during wet climatic phases that made it habitable and even hospitable. The sea level was lower, enough lower at certain periods that the strait separating East Africa from the Arabian coast was narrower than it is today, perhaps no more than 11 kilometers at its tightest point. It was through that strait, or something close to it, that the founders of all non-African humanity are believed to have crossed. The number of people involved in this crossing is a question that has consumed population geneticists for years. The genetic evidence, the sharply reduced diversity of non-African genomes compared to African ones, and the specific pattern of that reduction suggests that the founding population was very small. Estimates range from a few hundred breeding individuals to a few thousand. Some models suggest the effective population size, the number of individuals actually contributing genes to future generations, may have been as low as a few dozen at certain critical bottleneck moments. The uncertainty is large, but the consensus is clear. The founding group was tiny. The diversity they carried was a small fraction of the diversity present in Africa. And everything built since, every population on Earth outside that continent, grew from that compressed beginning. What followed was not a march. It was a drift. Generation by generation, groups moved slightly further along coastlines, slightly deeper into river valleys, slightly higher into mountain foothills. In each generation, the individuals making these small movements had no awareness of where their descendants would eventually be. A family group moving 20 kilometers east of their parents' campsite was not colonizing Asia. They were simply going where the food was. And then their children moved 20 kilometers further east. And their children's children. And across 1,000 generations, that accumulating drift became the settlement of the world. The speed of this process, when viewed across deep time, is astonishing. Genetic evidence suggests that modern humans reached southern Asia within a few thousand years of leaving Africa. An almost geologically instantaneous expansion. Australia was occupied by 65,000 years ago, a fact that requires either earlier than expected dates for the initial exodus or a rate of coastal movement that researchers find startling. Europe was reached from multiple directions, East Asia filled in from the south and west simultaneously. The Americas were the last major landmass to be occupied, with the first Americans crossing the Bering Land Bridge sometime between 25 and 15,000 years ago. But once they arrived, their expansion southward to the tip of South America required only a few thousand additional years. Throughout all of this movement, geography imposed a structure that was not chosen but was nonetheless systematic. Mountains funneled populations into valleys. Deserts deflected movement along their margins. River systems served as highways, enabling faster penetration of continental interiors than coastal routes could provide. The ocean was, for most of this period, the ultimate barrier. impossible except at its narrowest points, or, eventually, when the technology of watercraft made it bridgeable. Each of these geographic constraints functioned as a filter on the genetic diversity being carried forward. Every time a population was forced through a narrow passage, through a mountain pass, across a short water crossing, along a thin coastal corridor, only a subset of the total genetic variation made it through. The rest was left behind, either in the populations that stayed or in the lineages that did not survive the crossing. Every bottleneck reduced the signal. Every new territory was colonized by a founding population carrying a compressed version of what had come before. In machine learning, this process has a name. When you deliberately reduce the information flowing through a network, forcing it to compress its representation of the world, you call it regularization. The effect of regularization is, paradoxically, often an improvement. A network that has been forced to work with less information frequently generalizes better, adapts more readily to new situations, and performs more robustly under conditions it has never encountered before. The human network, it turns out, was being regularized continuously from the moment it stepped out of Africa, whether it knew it or not. When population geneticists began applying the mathematical tools of network science to the ancient migration data, they were not initially looking for a parallel with artificial intelligence. They were looking for a cleaner way to visualize and analyze the enormous complexity of human population history, to impose some order on the tangle of admixture events, dispersal routes, and divergence dates that the genome record was generating in bewildering quantity. Network science offered a natural framework. Instead of thinking about populations as points on a family tree, you could think of them as nodes in a graph, each one connected to others by edges that represented genetic relatedness, migration history, or both. The strength of each edge could be weighted by the degree of genetic similarity between the connected populations. The resulting graph was not a tree. It had loops and clusters and multiple pathways between the same pair of nodes. But it had structure, and that structure turned out to be highly informative. The most significant structural feature of the ancient human migration network is one that network scientists call scale-free topology. In a scale-free network, the distribution of connections across nodes follows a specific mathematical pattern. Most nodes have very few connections, but a small number of nodes, the hubs, have very many. The internet is scale-free. The network of airline routes is scale-free. The network of citations in scientific literature is scale-free. And, as the analysis of ancient migration data has now demonstrated with increasing rigor, the human population network is scale-free. The hub populations in the migration network are those that occupied critical geographic positions at key moments in human expansion, populations that served as the source of multiple subsequent dispersals, contributing genetic material to many different successor groups across many different directions. The Levantine population that served as the source of European colonization. the Central Asian population that fed dispersals both east into the Americas and west into Europe. The ancestral oceanic population that launched the extraordinary Polynesian expansion across the Pacific. These were not necessarily the largest or the most successful populations in their own right. They were simply the ones positioned at the nodes, the junction points where routes converged and from which multiple branches departed. The barriers in the migration network are equally important. The Sahara Desert did not merely slow movement between sub-Saharan and North Africa. It partitioned the network, creating a near total separation between the two sides that lasted for thousands of years and produced the deepest genetic divergence found anywhere in the human species. The Himalayan range created a similar partition in Asia, separating South Asian lineages from East Asian ones. The open Pacific created a partition that kept the Americas isolated from Eurasia for 60,000 years after initial colonization, allowing the independent development of genetic lineages and cultural traditions that, when they were eventually reconnected, proved to be among the most genetically distinct populations on Earth. In the language of computer networks, these barriers were firewalls, not deliberate, not programmed, but functionally equivalent. They controlled the flow of information, in this case, genetic information, between segments of the network, maintaining the integrity of isolated subsystems long enough for them to develop their own distinct internal structure. When the barriers broke, the effects were dramatic. The end of the last ice age opened corridors that had been closed for millennia, allowing populations that had been isolated to flood into contact with each other. The resulting admixture events, which the ancient DNA record has now documented in extraordinary detail, produced populations whose genetic composition was a blend of previously separate lineages, each one carrying distinct adaptations and distinct histories into a new combined system. A researcher at the Santa Fe Institute published a paper in 2017 demonstrating that when you draw the human migration network as a formal graph and compute its key structural metrics, degree distribution, clustering coefficient, average path length, and modularity, the resulting values are statistically indistinguishable from those produced by a trained deep neural network of comparable scale. The two networks, separated by every possible difference in substrate and timescale and purpose, occupy the same region of network space. They are, in the formal mathematical sense, topological twins. This was not the finding that the paper set out to report. It was a result that emerged from analysis the author had undertaken for a completely different reason. And it sat, largely unremarked, in the supplementary materials of a journal that most AI researchers never read. There is a property that network scientists value above almost all others: robustness. A robust network is one that continues to function, to move information from one part of itself to another, even when significant portions of it are damaged or destroyed. The internet is robust because it was designed to be any single node can fail without isolating the rest of the network, because information simply reroutes around the failure point. The brain is robust for the same reason. Even substantial damage to local regions rarely eliminates functions entirely, because those functions are distributed across multiple overlapping pathways rather than concentrated in any single one. The ancient human migration network is also robust, strikingly so, given that it was not designed by anyone. Multiple populations have been effectively destroyed over the course of human history, by disease, by ecological collapse, by conquest, without eliminating the genetic lineages they carried, because those lineages were also present in related populations elsewhere in the network. The population of ancient Britain was largely replaced by incoming Bronze Age farmers, but the hunter-gatherer genetic lineages they carried had already dispersed into Scandinavia and Iberia, where they survived. The ancient Tasmanian population, cut off from the mainland for 12,000 years, was destroyed by European colonization in the 19th century. But the lineages they carried were represented in mainland Australian populations who had diverged from them much earlier. What makes the migration network robust is the same property that makes it scale-free: the existence of hub populations that are connected to many other populations simultaneously. If any single hub is destroyed, the other hubs maintain connectivity across the network. Information, genetic information, keeps flowing. The diversity that accumulated over thousands of years of independent evolution is not lost. It persists in the distributed structure of the network itself. This is not unique to human migration. It is a general property of scale-free networks operating under pressure, and it shows up in every domain where such networks have been studied. Social networks are scale-free and robust. Ecosystems are scale-free and robust. Protein interaction networks inside cells are scale-free and robust. The property appears to be a near-universal consequence of the way networks grow when new nodes preferentially attach to already well-connected nodes. A growth rule that describes, with eerie accuracy, the way new human populations settled in relation to existing ones. And then there are the neural networks. When neuroscientists map the connectivity of the human brain at the level of regions, examining which brain areas are connected to which others, how strongly, and through how many pathways, they find a scale-free network with hub regions that are densely connected to many others, and peripheral regions with sparse, specialized connections. When AI researchers train deep neural networks on large data sets and then map the connectivity of the learned weight matrices, they find scale-free networks with hub nodes that process general features shared across many input types and peripheral nodes that process specific, narrow features. When population geneticists map the ancient migration network as described in the previous section, they find a scale-free network with hub populations that contributed genes to many subsequent groups and peripheral populations that remained isolated and specialized. 3 networks, 3 completely different substrates, 3 completely different timescales, 3 completely different purposes, and the same mathematical signature running through all of them. At this point in the investigation, 2 responses are possible. The first is to note the resemblance, file it under coincidence, and move on. The second is to ask whether all 3 systems were, in some fundamental sense, solving the same problem, and whether the shared mathematical signature is the mark left by that shared problem on every system that has ever faced it. The second response leads somewhere. The story that follows it is the story this documentary intends to tell. In 1943, a neurologist and a logician sat down together and wrote a paper that described, in abstract mathematical terms, how a neuron works. Warren McCulloch and Walter Pitts were not trying to build a computer. They were trying to understand the brain. Specifically, to understand how a system made of biological cells, each one capable only of simple electrochemical reactions, could collectively produce something as complex and flexible as thought. Their answer was elegant and, at the time, radical. The neuron is a threshold device. It receives inputs from many sources, weighs them and fires, passes a signal to the next neuron. When the combined weight of its inputs crosses a certain level, below the threshold, silence. Above it, signal. This simple rule, applied to a network of millions of interconnected units, could in principle produce any logical function that could be computed at all. The brain was not a special case. It was an instance of a general principle, that complex, adaptive behavior could emerge from a network of simple connected units, each following a simple rule. This was the founding insight of neural network research, and it opened a path that would lead, 70 years later, to systems capable of recognizing faces, generating language, diagnosing cancer, and playing games at levels no human could match. The artificial neural network that emerged from this tradition is architecturally simple. It consists of layers, typically 3 or more, of connected nodes. The first layer receives the raw input, numbers representing pixels in an image, frequencies in a sound, tokens in a text. Each node in this input layer passes its value to every node in the next layer, but not equally. Each connection carries a weight, a number that scales the signal being passed. A weight of 0 means the connection carries nothing. A weight of 2 means it doubles the signal. A negative weight means it inverts it. The weighted signals arriving at each node in the second layer are summed, passed through a transformation function, and sent onward to the third layer. This process repeats through every layer until the final one, which produces the output, a classification, a prediction, a generated text, an action recommendation. At each step, The calculation being performed is simple arithmetic. The complexity of what the system can do arises not from the complexity of any individual calculation, but from the sheer number of calculations performed simultaneously and in sequence, each one building on the results of the ones before it. This is worth sitting with because it is counterintuitive. There is no instruction in the system that says how to recognize a cat. There is no rule that defines what a face looks like or what constitutes a grammatical sentence or when a chess position is advantageous. The knowledge is not stored anywhere as a statement. It is distributed across the weights, encoded in the collective pattern of 2/10 here and -1.5 there, and 0.7 across a billion different connections, in a form that is not readable by any human analyst and cannot be translated into language, but that produces the right answer when tested. The way the network arrives at those weights is through learning. It is shown an input. It produces an output. The output is compared to the correct answer, and the discrepancy between what the network said and what it should have said is used to adjust the weights, slightly, carefully, in a direction that makes the error smaller. This process is repeated millions of times across millions of examples until the network's performance stabilizes at a level the designers consider acceptable. A neural network does not follow a program, it discovers one. The difference between these two things is the difference between a rule that was written and a rule that was found. And finding rules in noisy, complex data rather than being given them is precisely the kind of intelligence that evolution has been producing for billions of years. The resemblance at this level of description is not yet a mathematical argument, but it is a direction. The process by which a neural network adjusts its weights in response to error is called backpropagation, and it is, in a precise technical sense, the mechanism that makes modern AI possible. Without it, neural networks would be little more than sophisticated noise filters. With it, they become systems capable of mastering domains that were, until very recently, considered the exclusive province of human expertise. Here is how it works without the mathematics. The network makes a prediction. The prediction is wrong. Say, the network identified an image of a dog as a cat. The error, the gap between the prediction made and the correct answer, is calculated at the output layer. That error is then traced backward through the network, layer by layer, all the way to the input. At each layer, the question asked is, which connections contributed most to this error? The ones that contributed more are adjusted more. The ones that contributed less are adjusted less. The adjustment is made in the direction that would have reduced the error. Not to zero, because the network cannot know the correct answer in advance, but by a small amount determined by the learning rate, a parameter that controls how boldly the system moves in response to each mistake. After millions of repetitions of this process, forward pass, error calculation, backward adjustment. The weights settle into a configuration that minimizes the total error across the training dataset. At that point, the network has learned. The knowledge is in the weights, distributed across billions of connections, and the network can be presented with inputs it has never seen before and perform on them with remarkable accuracy. But there is a danger in this process, and understanding it is essential for understanding both neural networks and, as it will later emerge, human evolution. The danger is called overfitting. A network that is trained long enough on a fixed dataset can memorize the training examples rather than learning the underlying patterns they represent. It becomes extraordinarily good at the specific inputs it has seen and catastrophically bad at anything else. It has, in the language of machine learning, overfit its training data, become so precisely tuned to the signal it was given that it has lost the ability to generalize. The solution to overfitting is regularization, a family of techniques that deliberately introduce constraints or noise into the training process to prevent the network from becoming too dependent on any specific feature of the training data. The most powerful and widely used regularization technique is called dropout. During training, at each step, a random fraction of the network's neurons are temporarily disabled, set to 0, as if they do not exist. The network must learn to perform its task without them. When a different random fraction is disabled in the next step, it must perform without those instead. Over many training iterations, the effect is to prevent the network from developing fragile dependencies on specific neurons or pathways, forcing it instead to develop robust, redundant representations that are spread across many different parts of the network simultaneously. The result of dropout training is a network that is, in a very specific sense, stronger for having been weakened repeatedly. Its performance on new, unseen data is consistently better than the performance of networks trained without it, even though the individual training steps were harder. The constraint created capability. The third concept relevant to understanding neural network learning is weight initialization, the question of what values the weights are given before any training begins. This might seem trivial. If the network is going to learn the right weights through training anyway, why should the starting values matter? The answer, discovered through years of painful experience, is that they matter enormously. Networks initialized with very small random weights frequently fail to learn at all, a problem called vanishing gradients, in which the error signal that should propagate backward through the layers diminishes to nothing before it can influence the early layers. Networks initialized with very large random weights often explode. The adjustments grow uncontrollably, and the learning process diverges rather than converging. The starting condition shapes the entire trajectory. This is worth remembering. In both neural networks and, as it will emerge, in biological populations, where you begin determines, in ways that are not always obvious, where you can go. Step back from the mechanics of any individual neural network and look at the class as a whole. Look at every successful deep learning system built in the past 2 decades. The image classifiers, the language models, the game-playing agents, the protein structure predictors, and ask, what do they have in common? Structurally, despite their enormous differences in application and architecture. The answer is surprisingly consistent. First, they are all scale-free at the level of their learned weight structure. After training, the distribution of connection strengths across the network follows the same power law pattern found in scale-free networks. Most connections are weak, but a small number are very strong. And it is those strong connections, the hubs in the weight matrix, that carry the majority of the information. Second, they are all hierarchical. Early layers respond to simple generic features, edges in images, common word sequences in text, while later layers respond to increasingly complex and specific combinations. The complexity is not programmed in, it emerges from the training process. Third, they are all robust. Trained neural networks can have a substantial fraction of their neurons removed or their weights corrupted without catastrophic loss of performance. The function is distributed, redundant, and resilient. These 3 properties, scale-free connectivity, hierarchical feature extraction, robustness through redundancy, are not independent choices made by neural network designers. They are emergent consequences of the training process. Properties that arise whenever a sufficiently large network is trained on sufficiently complex data with appropriate regularization. They emerge because they are the structural signature of a system that has learned to generalize, to capture the genuine deep patterns in its training data rather than the superficial, incidental ones. Now recall what Chapter 3 established about the ancient human migration network. It is scale-free. Hub populations at critical geographic junctions are connected to many successor groups. The vast majority of populations have only a few connections. The degree distribution follows a power law. It is hierarchical in the sense that large regional groupings divide into continental groupings, which divide into local ones, with different levels of the hierarchy encoding different scales of shared ancestry. And it is robust, demonstrably capable of maintaining genetic continuity even through events that destroyed large fractions of the network. Scale-free, hierarchical, robust. The human migration network and the trained neural network share all 3 defining structural properties. This is the point in the investigation where the comparison stops being an observation and starts becoming an argument. The argument is this: both systems are solutions to the same problem. The problem is how to move information. Genetic information in one case, numerical information in the other, from a source to a destination across a complex, noisy, unpredictable environment in a way that preserves signal, adapts to new inputs, and survives local failures. This problem has specific mathematical constraints, and systems that solve it well tend to converge on the same class of architectures. the same scale-free, hierarchical, robust network topology, regardless of their substrate. The convergence is not a coincidence. It is evidence that the solution space for this class of problem is limited, that there are only a few ways to solve it well, and that both biological evolution and artificial neural network research have independently found the same ones. The question that remains, the question that drives the next several chapters, is how deep the correspondence goes. Is it merely structural, a matter of topology, or does it extend into the dynamics of learning itself, into the specific mechanisms by which both systems improve their performance, adapt to new environments, and transmit what they have learned? If the structure is the same, are the processes also the same? And if the processes are the same, what does that tell us about the nature of intelligence? About what it is, at its most fundamental level, to learn? Scientific fields have a tendency to build walls, not deliberately but structurally, through the accumulation of specialized vocabulary, specialized methods, specialized publication venues, and specialized professional identities that make it genuinely difficult for a researcher in one area to follow the work being done in another. Population genetics and machine learning are both technically demanding fields with rich internal literatures and their own distinct cultures. The people who work in one rarely read the journals of the other. This is partly why the correspondence between the two systems went largely unnoticed for as long as it did. Each piece of the puzzle was available. The scale-free topology of the migration network had been documented in population genetics journals. The scale-free topology of trained neural networks had been documented in computational science journals. The mathematical equivalence between natural selection and stochastic optimization had been noted in passing in theoretical biology papers. But no single paper had brought these findings together and asked whether they constituted evidence of a deeper structural identity rather than a collection of parallel observations. That synthesis, when it finally began to appear in the literature, came not from either parent field, but from the borders between them, from computational biology, from network science, from theoretical evolutionary biology. A handful of researchers, working in the spaces between disciplines, began to notice that the tools they were importing from machine learning to analyze genetic data were not merely convenient. They were producing results that described the genetic data as naturally as if the tools had been built for it, because in a sense that was becoming harder and harder to dismiss as coincidence. The genetic data and the machine learning systems were both instances of the same underlying mathematical structure. The convergence operates at 3 levels, each one more fundamental than the last. At the topological level, both systems are scale-free networks with the same structural metrics. This is the level of resemblance, the two maps that look the same. At the mechanistic level, both systems use structurally equivalent processes to learn and adapt. Population bottlenecks correspond to dropout regularization. Gene flow corresponds to backpropagation. Founder effects correspond to weight initialization. This is the level of function. The two systems not only look alike, but work alike. At the mathematical level, both systems are instances of stochastic gradient descent on a high-dimensional optimization surface, the same class of algorithm, running in different substrates across different time scales, finding the same class of solution. This third level is the one that matters most, and it is the one that the remaining sections of this chapter will build toward. But it is useful to begin at the first level, because the topological resemblance is the entry point, the observation that demands explanation and drives the investigation toward the mechanism. Everything that follows is, in one sense, an answer to the question posed by those two maps. 3 specific correspondences will now be examined in sequence. Each one is a relationship between a mechanism of neural network learning and a mechanism of human population genetics. Each one is supported by documented evidence, and together they make a case that is difficult to explain away as coincidence. Section 5.2 Population bottlenecks and dropout regularization. Return to the dropout mechanism described in the previous chapter. A neural network is being trained. At each training step, a random fraction of its neurons, typically between 20 and 50%, are switched off. The network must complete its task with a reduced complement of resources, relying on the neurons that remain to carry the full weight of representation. In the next step, a different random fraction is switched off, and the next, a different one still. The effect of this repeated random deprivation is counterintuitive and profound. A network trained with dropout does not develop deep dependencies on any specific neuron or pathway. Because any neuron might be absent on any given step, every neuron must be useful independently, must encode information in a form that does not rely on any other specific neuron being active to make sense. The result is a network whose representations are distributed, redundant, and robust. Remove any individual neuron from a dropout-trained network, and the performance barely flickers. Remove the same neuron from a network trained without dropout, and the damage can be severe. Now consider a human population experiencing a severe bottleneck. A disease, a famine, a volcanic winter. Some catastrophe reduces the population from tens of thousands of individuals to a few hundred or a few dozen. The genetic diversity carried by the pre-bottleneck population, the accumulated variance representing thousands of generations of adaptation to local environments, is not transferred intact. Only a fraction of it survives, carried in the genomes of the survivors. The fraction that survives is not selected for its quality. It is selected by chance. The survivors are not the most diverse individuals in the original population. They are simply the ones who happened to be alive when the catastrophe passed. The resulting post-bottleneck population is genetically impoverished compared to its predecessor. It has fewer variants, less diversity at most genetic loci, and a reduced capacity for fine-grained local adaptation. In the short term, this impoverishment is a liability. But in the long term, across the hundreds or thousands of generations that follow, something unexpected happens. Populations that have passed through severe bottlenecks frequently show exceptional capacity for rapid adaptation to new environments. The classic examples are the founding populations of the Americas and the Pacific, both of which passed through dramatic bottlenecks and both of which subsequently demonstrated extraordinary adaptive flexibility, colonizing environments as diverse as Arctic tundra, tropical rainforest, high-altitude steppe, and open ocean. The mechanism behind this counterintuitive finding is exactly the one that makes dropout effective in neural networks. A post-bottleneck population, having lost the deep local specializations of its ancestor, is not locked into any specific adaptive strategy. Its genetic architecture, simplified by the bottleneck, is more plastic, more capable of rapid reconfiguration in response to new selection pressures. The compression enforced by the bottleneck has, paradoxically, increased its capacity for generalization. Both systems, the neural network and the human population, become more capable by becoming simpler. The catastrophe is not a setback from which they recover. It is a regularization event from which they emerge changed. The loss of complexity at the level of individual components produces a gain in robustness and adaptability at the level of the system as a whole. And this is not a metaphor. It is the same mathematical relationship operating through structurally equivalent mechanisms in two systems that have never been in contact. Backpropagation works by taking the error at the output, the gap between what was predicted and what was true, and distributing responsibility for that error backward through the network, adjusting each connection according to its contribution. The correction does not remain local. A mistake at the output changes connections deep in the interior of the network, in layers that may be many steps removed from the point where the error was first calculated. The adjustment propagates backward, modifying the global architecture of the network in response to local feedback. Gene flow is the movement of genetic material from one population to another through interbreeding between previously separated groups. When two populations that have been evolving independently come into contact and mate, the genetic information carried by the incoming population does not stay at the point of contact. It propagates forward through subsequent generations, spreading through the recipient population at a rate governed by natural selection and random drift, ultimately reaching a frequency that reflects its adaptive value to the recipient. If the incoming variants improve survival and reproduction, they spread quickly. If they are neutral, they spread slowly and randomly. If they are harmful, they diminish. The parallel with backpropagation is structural and, it turns out, mathematically precise. The incoming genetic information corresponds to the error signal, not because the information represents a mistake, but because it represents information from a different source that modifies the existing architecture of the recipient system. Just as backpropagation adjusts connection weights throughout the network in proportion to their contribution to the output error, natural selection adjusts allele frequencies throughout the recipient genome in proportion to their contribution to overall fitness. In both cases, the adjustment is global rather than local. In both cases, it is proportional rather than uniform, and in both cases, it propagates backward through the existing structure, modifying connections that were established long before the new information arrived. The most instructive example of this process is also the most dramatic in human evolutionary history. When modern Homo sapiens populations moved out of Africa and encountered Neanderthals in Europe and Western Asia, the two species interbred. The Neanderthal genetic material that entered modern human populations was not uniformly distributed across the genome. Certain Neanderthal variants were rapidly favored by selection and reached high frequency in modern human populations, particularly variants related to immune function and skin adaptation to lower light environments. Other Neanderthal variants were rapidly eliminated, particularly those that appeared on the X chromosome, where the interaction with modern human genetic architecture produced adverse effects. This is a backpropagation event. The information from the Neanderthal genome entered the modern human network, propagated through it under the selective pressure of the recipient environment, and modified the global architecture of the recipient system in ways that improved its performance, its survival and reproduction in its current environment. Certain connections were strengthened, others were weakened. The network, after the event, was measurably different from the network before it, and measurably better adapted to the environment it occupied. The Neanderthal introgression was not the only such event. The ancient DNA record now documents dozens of similar gene flow events across human prehistory, each one a backpropagation step, each one a moment when information from outside the existing architecture entered and modified it, improving the fitness of the recipient system in ways that pure internal evolution could not have achieved in the same time frame. The third correspondence is the subtlest and perhaps the most philosophically resonant of the three. Before a neural network sees a single piece of training data, its weights must be initialized, given starting values that will be adjusted during training. For decades, researchers used simple random initialization, assigning each weight a small random number drawn from a normal or uniform distribution. The network's subsequent learning then adjusted these initial values towards something useful. It seemed to follow, reasonably, that since all the initial values would eventually be overwritten by training, their specific starting values should not matter much. Experience proved this assumption wrong. Networks with different initialization strategies trained on identical data with identical architectures and hyperparameters, converged to reliably different solutions. Some initializations produced networks that learned quickly and generalized well. Others produced networks that learned slowly, converged to poor solutions, or failed to learn at all. The starting condition cast a shadow over the entire learning trajectory. Where the network began determined in ways that could not be fully anticipated or controlled, where it was capable of going. The founder effect in population genetics is precisely this phenomenon translated into biological terms. When a small group breaks away from a larger population and establishes a new one in a new territory, the genetic composition of that founding group determines the genetic architecture of all subsequent generations in that lineage. The founders are not a representative sample of the source population. They are a chance sample, a random subset carrying a random fraction of the source population's total diversity. Rare variants that happen to be present in the founders may become common in the descendant population simply because there was no one else to inherit from. Common variants from the source population that happen not to be present in the founders may be absent from the descendant lineage for thousands of years or permanently. The Ashkenazi Jewish population provides a particularly well-documented example. Founded from a small group of individuals in medieval Europe, the population has since expanded to millions, but it carries elevated frequencies of several rare genetic variants that were present by chance in the founders, variants associated with conditions that are rare in other populations. The founder event initialized this population with a specific genetic architecture, and the weight of that initialization has persisted measurably and consequentially for over 1,000 years. The lesson of both systems is the same. Starting conditions are not overwritten by subsequent experience. They are built upon. They shape the space of possible futures, and they do so not through intention or design, but through the simple, implacable logic of a system that cannot go back and start over. It can only continue from where it is, carrying everything that came before in the structure of what it has already become. There is a moment in the human genetic record that looks unlike anything else. A point of such severe compression, such near total reduction, that the diversity surviving on the other side of it is almost vanishingly small compared to what must have existed before. Scientists call it the human bottleneck, and they place it somewhere between 60 and 80,000 years ago. The cause is debated. The evidence for the reduction itself is not. The leading candidate for the cause is a volcano. On the island of Sumatra, in what is now Indonesia, sits the caldera of a supervolcano called Toba. Approximately 74,000 years ago, Toba erupted on a scale that dwarfs anything in recorded human history. Dwarfs, in fact, anything in the last 2 million years of geological record. The eruption ejected an estimated 2,800 cubic kilometers of material into the atmosphere. Ash fell to depths of several meters across South and Southeast Asia. Sulfur dioxide injected into the stratosphere created a reflective layer that reduced solar radiation reaching the Earth's surface, triggering a volcanic winter that persisted for years and possibly decades. The effect on human populations, already small and scattered, would have been catastrophic. Archaeological evidence from sites in India shows a sharp discontinuity at the Toba ash layer. Rich assemblages of tools above and below, but the specific tool-making traditions above the ash are different from those below, suggesting that the populations who created them were different. That the pre-Toba inhabitants of many regions were largely eliminated and replaced by survivors or migrants from elsewhere. The genetic evidence tells a consistent story. Non-African human populations show far less diversity than African ones, and the date at which the diversity reduction occurred aligns with the Toba eruption. But here is the part of the Toba story that rarely makes it into popular accounts, because it seems at first to contradict everything that should follow from near extinction. The survivors, whatever small remnant of humanity made it through the volcanic winter, did not merely continue. They expanded. With astonishing speed, by the slow standards of deep time, the post-Toba human population grew, diversified, and spread. Within 10,000 years of the eruption, modern humans had reached Australia. Within 20,000 years, they had colonized much of Asia. The near extinction preceded an explosion. This pattern, catastrophic reduction followed by rapid adaptive radiation, is not unique to the Toba event. It appears again in the colonization of the Americas, in the founding of Pacific Island populations, in every major bottleneck event in the human genetic record. The reduction does not destroy the capacity for expansion. It appears, paradoxically, to enhance it. The neural network parallel is exact. A network that has been trained through aggressive dropout, repeatedly forced to function with a reduced complement of resources, consistently outperforms a network trained without such deprivation when both are tested on new, unseen data. The constraint imposed during training translates into capability during deployment. The impoverishment during the learning phase translates into richness during the application phase. Toba was, in the language of machine learning, an extreme regularization event. It stripped the human network of nearly everything, nearly every variant, nearly every lineage, nearly every local adaptation that had accumulated across the preceding hundreds of thousands of years. What remained was the minimum viable signal, the small compressed core of human genetic information that was sufficient to regenerate a functional, adaptable, and ultimately extraordinarily capable population. There is something deeply uncomfortable about this framing, and it is worth acknowledging directly. It does not diminish the enormity of what was lost. It does not make the catastrophe anything other than a catastrophe for the individuals who lived and died through it. But at the level of the network, the system as a whole, viewed from the outside across geological time, the Toba bottleneck did not end the human story. It simplified it, and that simplification, that compression, created the conditions for everything that followed. 12,000 years ago, the Bering Land Bridge, Beringia, was a substantial landmass connecting what is now Siberia to what is now Alaska. It was not a narrow isthmus. At its widest, it stretched over 1,500 kilometers from north to south, a subcontinent in its own right, inhabited by people who may have been there for tens of thousands of years before the glacial passages opened and allowed expansion southward into the Americas. The genetic record of indigenous American populations tells a story of unusual compression. Of all the major human populations outside Africa, Americans show the lowest mitochondrial DNA diversity, a level of genetic homogeneity that reflects both the severity of the original bottleneck and the additional isolation imposed by the closing of the Beringian corridor as glaciers re-advanced. The founders of the Americas were not a broad cross-section of Siberian genetic diversity. They were a subset of a subset, a small population that had itself descended from a small founding group, which had itself been shaped by the earlier bottlenecks of the out-of-Africa migration. What this population did with that compressed starting point is one of the most remarkable adaptive stories in the human record. Within 2,000 years of the opening of the ice-free corridor, humans had reached the southern tip of South America, more than 15,000 kilometers from the Beringian entry point. Along the way, they adapted to environments ranging from the boreal forests of North America to the subtropical deserts of the Southwest, to the high-altitude grasslands of the Andes, to the temperate rainforests of Patagonia. They developed independently agricultural traditions in at least 3 distinct centers: the highlands of Mexico, the highlands of the Andes, and the tropical lowlands of Amazonia. They built cities, engineered water systems, and developed calendrical and mathematical traditions of significant sophistication. All of this from a founding population that, genetically, was among the least diverse of any human group on Earth. The Polynesian case is even more extreme. The ancestors of the Polynesian peoples were themselves already the product of multiple bottleneck events. The expansion out of Taiwan roughly 4,000 years ago, the crossing of island Melanesia, the settlement of Fiji and Tonga. Each crossing was a bottleneck. Each island founding group was a compressed subset of the group that had come before. By the time Polynesian navigators reached the most remote islands of the Pacific, Hawaii, Easter Island, New Zealand, the founding populations were sometimes small enough to count in dozens, and yet the cultures that developed from these miniature founding groups were, by any measure, extraordinary. Polynesian navigation technology, the ability to sail thousands of kilometers across open ocean using stars, swells, and birds without instruments, with consistent success, represents one of the most demanding cognitive and technical achievements in pre-industrial human history. Polynesian agricultural systems adapted to island environments with remarkable speed and ingenuity. Polynesian social and political organization developed forms of institutional complexity that European observers consistently underestimated. The machine learning researcher looking at these cases would recognize the pattern immediately. In the experimental literature on neural network initialization, there is a well-replicated finding. Networks initialized from a compressed representation of a previously trained network, a kind of distillation of what a larger system learned into a smaller architecture, consistently outperform networks of the same size trained from scratch on the same data. The compression enforced by the smaller architecture forces the distilled network to capture only the most essential, most general features of the task, discarding the specific and the incidental. The result is a network that generalizes better than either the large original or a small network trained naively. The Beringian founders and the Polynesian navigators were not distillations in any engineered sense. But the process they went through, the repeated compression of genetic diversity through successive bottlenecks, each one forcing the surviving system to rebuild on a smaller and simpler genetic foundation, produced something functionally equivalent. Systems shaped by constraint, tested by scarcity, rebuilt from minimal starting conditions, and then released into complexity and found repeatedly to be more than equal to it. A natural question arises from the bottleneck evidence. If the compression produced by a severe population reduction is genuinely beneficial at the network level, what exactly is being preserved through the compression? What survives a catastrophic bottleneck that is worth keeping? And what is lost that does not turn out to matter? The answer is not what classical evolutionary theory would predict. The classical prediction is that bottlenecks are unambiguous disasters, that the reduction in diversity is a reduction in the raw material that natural selection needs to operate, and that any population emerging from a severe bottleneck is, everything else being equal, less capable of adapting to future challenges than the ancestral population it descended from. This prediction is broadly correct in the short term. In the long term, the evidence suggests a more complex picture. What tends to survive bottlenecks at elevated frequencies are genetic variants associated with broad, general-purpose adaptive functions. Variants related to immune flexibility, the capacity to respond to novel pathogens rather than optimizing for specific known ones. Variants related to metabolic generalism, the ability to extract energy from diverse food sources rather than thriving optimally on a specific diet. Variants related to behavioral plasticity, the neurological architecture that supports learning, novelty-seeking, and social cooperation, which are the traits most useful in unpredictable and rapidly changing environments. What tends to be lost at high rates during bottlenecks are variants associated with narrow, specific adaptations. fine-tuned local immunities, metabolic specializations that require specific foods, physiological adaptations to specific altitudes, specific climates, specific ecological niches. These are the variants that were, in the pre-bottleneck environment, the most immediately useful, the best fit responses to the specific conditions of the place and time. They are also the most fragile, the most dependent on the continuation of the specific conditions that made them valuable, and the least useful in the new and unknown environments into which post-bottleneck populations inevitably expand. There is a concept in information theory called minimum description length. It refers to the shortest code that can fully represent the meaningful content of a dataset, the most compressed form of the signal that retains everything important and discards everything redundant. A bottleneck event in the population genetics context functions as a natural minimum description length operation. The catastrophic reduction strips away the specificity and the redundancy, leaving behind a compressed representation that contains the essential structure, the general purpose, flexible, broadly adaptive core of the system that came before. Neural network researchers have shown repeatedly that compressed models, networks trained with aggressive regularization or distilled from larger models or pruned of weakly connected nodes, tend to learn representations closer to the minimum description length ideal. They learn the essential structure of their training data rather than the surface features. And that essential structure turns out to be more transferable to new domains, more robust to distribution shift, more useful in conditions that differ from the training environment. The human populations that emerged from the great bottlenecks were not the strongest populations. They were not the largest or the most genetically sophisticated. They were the most compressed, the most stripped of specialization, the most reliant on general purpose capabilities, the most dependent on the plasticity and learning capacity that, in the absence of specific inherited solutions, became the only tools available. And those tools, it turned out, were sufficient for everything. For navigating open ocean without instruments, for farming the Andes at 4,000 meters, for surviving the Arctic winter, for building cities, writing histories, composing music, and eventually, many tens of thousands of years after the first bottleneck, for sitting in a laboratory and mapping the genetic record of all the bottlenecks that came before. There is a landscape that no one has ever seen, but that every organism that has ever lived has been navigating. It has billions of dimensions. Its terrain is defined by a single quantity: reproductive success. The peaks of this landscape, the high points, represent combinations of genetic characteristics that produce few surviving offspring. The valleys, the low points, represent combinations that produce many. Natural selection is the force that pushes populations downhill toward the fertile valleys, generation by generation, guided by the imperfect gradient of fitness differences between individuals who vary slightly in their genetic makeup. This image is Sewall Wright's fitness landscape, first published in 1932 and still one of the most productive conceptual tools in evolutionary biology. It is a visualization device, a way of thinking about evolution as a process of movement through an abstract space of possibilities, guided by a fitness function that evaluates each position in that space. The visualization is imperfect because the actual fitness landscape has far too many dimensions to draw, and because the landscape itself changes as environments change. But as a conceptual scaffold, it is remarkably close to something that machine learning researchers re-derived independently decades later for completely different reasons. In machine learning, the equivalent of the fitness landscape is called the loss surface. It is the space of all possible parameter settings for a neural network, all possible combinations of all possible weight values, and the altitude at each point in that space represents the error made by the network when those particular weights are used to make predictions. Like the fitness landscape, it has vastly more dimensions than can be visualized. Like the fitness landscape, it is defined by a single evaluative quantity. In one case, reproductive success, in the other, prediction error. Like the fitness landscape, the goal is to move to the lowest possible point. Gradient descent, the algorithm that neural networks use to find their way to low points in the loss surface, works exactly as the name suggests. At each step, the algorithm calculates the slope of the loss surface at the current position. Which direction leads most steeply downhill? Then it moves a small step in that direction. The size of the step is controlled by the learning rate. The direction is determined by the gradient, the mathematical measure of how much the loss changes for a small change in each weight. Natural selection is, formally, gradient descent on the fitness landscape. The selection differential, the difference in reproductive success between individuals with different genetic variants, measures the slope of the fitness landscape at the current position of the population. The response to selection, the change in allele frequency that occurs over one generation, is the step taken in the direction of that slope. The heritability of the selected trait controls how large a step is taken, in much the way the learning rate controls step size in gradient descent. This formal equivalence was noticed and stated precisely by theorists working in the overlap between evolutionary biology and machine learning. And it is not a loose analogy. The same equations that describe one system, with the variables relabeled, accurately describe the other. The mean change in allele frequency under selection is governed by the same mathematical relationship as the mean change in network weights under backpropagation. The formal structure is identical. The most important consequence of this identity is that everything known about gradient descent its strengths, its failure modes, the techniques for improving its performance, applies in corresponding terms to natural selection. And everything known about natural selection, how it behaves in small populations, how it responds to environmental change, how it deals with the complex, multidimensional problem of optimizing fitness in a rugged landscape, applies in corresponding terms to gradient descent. Two fields separated by a century of independent development turn out to have been studying the same algorithm the entire time. The most dangerous feature of any fitness landscape or lost surface is the local minimum. A local minimum is a point that is lower than every neighboring point, but not the lowest point on the entire surface. A population or a network that reaches a local minimum faces a dilemma. The gradient at that point tells it to stay still because every direction is uphill. But staying still means settling for a solution that might be far from optimal, that might, in fact, be quite bad by global standards, merely not actively harmful by local ones. Evolutionary biology has documented local minimum traps with uncomfortable frequency. Species that evolved in stable, isolated environments, island species, cave species, populations of large animals with long generation times, frequently show evidence of being locked into adaptive strategies that work well in their current environment, but are brittle when that environment changes. The highly specialized beak of a particular bird, exquisitely optimized for a particular seed. The highly specific immune profile of a population that has never encountered a particular class of pathogen. The metabolic architecture fine-tuned to a single food source that may not always be available. These are local minima, solutions that are locally stable but globally suboptimal and from which the path to a better solution runs, unavoidably, uphill. Machine learning systems face the same problem. A neural network trained on data with a specific statistical distribution may converge to a set of weights that produce excellent performance on that distribution, but poor performance on slightly different inputs. It has found a local minimum in the lost surface, a configuration that satisfies the gradient descent algorithm by being locally flat. But that would not be the best solution achievable on a more diverse training set. The solution in both systems is noise. In machine learning, stochastic gradient descent, the version of gradient descent actually used in practice, adds randomness to the optimization process by computing the gradient not from the full training dataset, but from a small random sample of it. This means that the direction taken at each step is not the true downhill direction from the current point, but a noisy approximation of it. The noise allows the optimizer to occasionally move slightly uphill, to escape the immediate pull of a local minimum and explore the broader landscape in search of a deeper valley. In population genetics, the equivalent mechanism is genetic drift, the random fluctuation of allele frequencies in finite populations that occurs simply because reproduction is not a perfectly deterministic process. In a large population, genetic drift is a weak force, easily overwhelmed by selection. but in small populations. And small populations are precisely the ones that emerge from bottlenecks. Genetic drift is powerful enough to push allele frequencies significantly in random directions, sometimes moving a population away from a local fitness optimum and onto the slope of a distant, higher peak. The parallel is mathematically precise and practically consequential. Darwin's finches, the famous suite of beak shapes in the Galapagos, each optimized for a different food source on a different island, represent multiple local minima in the finch fitness landscape. The founding populations of each island were subject to genetic drift as well as selection, and the path each population took to its current adaptive peak was shaped by the random fluctuations of small founding groups as much as by the deterministic push of natural selection. Different random starting conditions, different drift trajectories, different local minima reached, producing the extraordinary morphological diversity that provided Darwin with his clearest evidence for evolution by natural selection. Antibiotic resistance in bacteria provides a more urgent demonstration. Bacteria evolving resistance to a new antibiotic do not follow a single deterministic path through the fitness landscape. They explore multiple paths simultaneously through the combination of rapid mutation and the stochastic dynamics of growth in small subpopulations. Some paths hit local minima, partial resistance that does not fully protect the organism. Others, by chance, reach the high ground, full resistance. The stochastic nature of the process is not a bug. It is the feature that allows evolution to explore a rugged landscape more efficiently than a purely deterministic optimizer ever could. Stochastic gradient descent in machine learning was invented because deterministic gradient descent failed to scale. Natural selection is stochastic because deterministic selection cannot navigate a rugged fitness landscape. Two fields solving the same problem, finding the same solution. There is a way of looking at the human genome that is unfamiliar and, at first, slightly disorienting. It is not the way biologists typically talk about DNA, and it is not the way the subject is taught in schools. But it is, in a certain technical sense, the most accurate description available. The human genome is a trained model. It is the output of a 3.8 billion year optimization process that has been performing gradient descent, noisy, stochastic, iterative, continuously updated gradient descent on a loss surface defined by reproductive success in a given environment. The training data is every environmental challenge the lineage has ever faced. Every pathogen, every climate shift, every change in food availability, every predator, every competitor, every partner. The trained weights are the allele frequencies in the current population. The specific distribution of genetic variants that encodes the accumulated solutions to every adaptive problem the lineage has survived. The architecture of the model, the genome structure, the organization of its coding and non-coding regions, the distribution of regulatory elements, reflects both the initial conditions of the lineage and the history of bottleneck events, admixture pulses, and selection pressures that have shaped it. Some regions of the genome are highly conserved across all human populations, reflecting solutions that were found early and have not been improved upon in 100,000 years of subsequent optimization. Other regions show high diversity across populations, reflecting ongoing optimization for the specific conditions of each local environment. Still others show the signatures of recent strong selection, rapid frequency changes in the past few thousand years, driven by the new environmental pressures of agriculture, urban living, and the diseases that accompany dense human settlement. The model is not finished. It is still running. Every generation adds new training data. Every birth and death is a measurement of current performance. Every generation in which certain variants produce more children than others is a gradient step, a small adjustment in the weights, pushing the population slightly closer to the local minimum of the current fitness landscape. The process is slow, its output is never stable, and the landscape itself is constantly shifting as environments change, as new diseases emerge, as human social structures alter the selection pressures operating on behavioral and cognitive traits. But the model has been running long enough to produce something remarkable. The human genome, the collective genetic architecture of the species, is a compressed, distributed representation of 8 billion years of adaptive solutions. Not 8 billion years of the human lineage specifically, but of the entire lineage of life from which humans descend. Each generation's genome inheriting the accumulated learning of all the generations that preceded it, and adding its own small increment of new information. When machine learning researchers speak of pre-trained models, large AI systems that have been trained on vast data sets and then adapted for specific tasks, they are describing something whose logical structure is very similar to what evolution has been producing all along. A large, general-purpose system shaped by extensive exposure to complex data, whose internal representations encode useful patterns that can be quickly adapted to specific applications without starting from scratch. The human brain is a specific application. It is a specialized module built on top of the general-purpose platform of the mammalian genome, which is itself a specialized module built on the general-purpose platform of the vertebrate genome, which descends from the general-purpose platform of the first eukaryotic cell. Each level of this hierarchy is a form of transfer learning, the application of previously trained representations to a new and more specific optimization problem. And at the top of this hierarchy, the most recent and most specialized module of all is the one that looked at the genome and said, "This looks familiar." Transfer learning is one of the most powerful ideas in modern machine learning, and it arrived as a solution to a specific practical problem. Training a large neural network from scratch on a complex task requires enormous quantities of data and computational resources. But many tasks are related. They share underlying patterns, overlapping feature spaces, common structural properties. A network trained to recognize objects in photographs has learned representations of edges, textures, shapes, and spatial relationships that are useful not just for object recognition, but for any visual task. Why rebuild those representations from scratch for every new application when they already exist, encoded in the weights of the trained network? Transfer learning takes the weights of a model trained on one task and uses them as the starting point for training on a different, related task. The new model does not start from random initialization. It starts from a configuration that already encodes useful general-purpose representations, derived from the extensive training of the source model. The amount of new training data required to reach good performance is dramatically reduced. The quality of the final model is often higher than could be achieved by training on the new task alone. Genetic admixture is transfer learning. When populations that have been evolving in isolation come into contact and interbreed, the genetic material exchanged between them carries encoded adaptive solutions. Allele frequencies that represent the accumulated learning of separate evolutionary histories, tuned to separate environments, solving separate versions of the common problem of survival. That encoded information does not arrive as raw data requiring processing from scratch. It arrives as a configuration, a set of variant frequencies that already represents a solution, however imperfect its fit to the recipient environment. The critical insight is that the incoming genetic material does not simply add to the recipient population's existing diversity. It interacts with it in the same way that a transferred model's weights interact with the new training objective. Variants from the incoming population that are compatible with the recipient's existing genetic architecture, that produce beneficial interactions when expressed alongside existing variants, are rapidly favored by selection and incorporated at high frequency. Variants that are incompatible, that produce harmful interactions with the existing architecture, are rapidly eliminated. The process is selective integration, a filtering of incoming information through the existing structure of the recipient system, retaining what is compatible and useful, discarding what is not. The result of this selective integration, documented now across dozens of well-characterized admixture events in human history, is consistently more complex than what either source population could have achieved alone. The ancestors of modern South Asians were formed by the mixture of an ancient hunter-gatherer population with an expanding agricultural population from the West, and the resulting admixed population shows metabolic and immune profiles distinct from both sources, adapted to the specific conditions of the South Asian environment in ways that appear to involve contributions from both ancestral groups. The ancestors of modern Europeans were formed by the mixture of three distinct source populations: ancient hunter-gatherers, early farmers from Anatolia, and steppe pastoralists from the Pontic grasslands. And the resulting genetic architecture shows signatures of selection on the combined population that are not present in any of the three sources individually. These are not metaphorical data merges. They are instances of information transfer between trained systems with selective integration of compatible parameters and rejection of incompatible ones, producing a combined system whose capabilities exceed those of either source. The mathematical structure of the process is formally equivalent to transfer learning with selective fine-tuning. The biological substrate is human bodies rather than silicon chips. The timescale is generations rather than epochs. But the operation being performed is, at the level of the mathematics, the same. A 2019 genomic study scanning ancient DNA samples from across Eurasia found that virtually every sampled population showed evidence of at least 4 and often 6 or more distinct admixture pulses. Separate events of incoming genetic information, each one a discrete transfer learning step, each one leaving a traceable signature in the resulting population's genetic architecture. The human genome is not the output of a single training run. It is the output of a long sequence of transfer learning events, each one building on what came before. One of the most powerful tools in a machine learning researcher's toolkit is also one of the most mathematically fundamental: principal component analysis. It is a method for finding hidden structure in complex data. Given a large data set with many variables, many dimensions, principal component analysis identifies the directions in that high-dimensional space along which the data varies most. It compresses the data onto those directions, revealing the underlying structure that was present in the original data, but was hidden by the noise and redundancy of all the other dimensions. Applied to the internal weight matrices of a trained neural network, principal component analysis reveals the learned representations, the features the network has discovered as most useful for its task. Applied to the output activations of different layers, it maps the information processing hierarchy, showing how raw inputs are progressively transformed into abstract representations as the data moves deeper into the network. Population geneticists, independently and for entirely different reasons, developed the same mathematical tool, also called principal component analysis, also applied to a high-dimensional data set, also looking for hidden structure, to analyze genetic variation across human populations. In their application, the dataset is a matrix of allele frequencies across thousands of genetic loci and hundreds of populations. The principal components that emerge from this analysis correspond with remarkable reliability to real features of the population's ancestral history. The first principal component often separates populations by continental ancestry. The second separates within continental groupings, and so on. Each successive component revealing progressively finer-grained structure in the data. When population geneticists first began applying this tool systematically in the early 2000s, the results were striking. The principal component maps of global genetic variation showed structure that corresponded closely to geographic structure. Populations from the same continent clustered together. Populations from adjacent continents were adjacent on the principal component map, and the overall shape of the variation was consistent with the pattern of human migration from Africa outward. The tool was not just finding statistical patterns, it was reading the actual history of movement encoded in the genetic data. With the addition of ancient DNA, the resolution of these analyses has become extraordinary. Modern populations are weighted superpositions of ancient source populations. Their allele frequencies reflect the contributions of multiple ancestral groups, each weighted by the fraction of ancestry that group contributed. The principal component analysis of a modern population's genetic data reveals these weights. It decomposes the observed allele frequency pattern into contributions from the ancient source populations, with each component corresponding to a specific ancestral group. The correspondence with neural network analysis runs deeper than analogy. When researchers apply principal component analysis to the learned representations of a deep neural network, they find that the representation of any given input can be decomposed into contributions from a set of basis vectors, learned features that capture the most important patterns in the training data. The input's representation is a weighted superposition of these basis vectors in exactly the way that a modern population's genetic composition is a weighted superposition of ancestral source populations. The mathematics is formally identical. The interpretation is different. Genetic ancestry in one case, learned features in the other, but the structure of the decomposition is the same. There are populations whose genetic composition is so thoroughly understood through this kind of analysis that researchers can state with quantitative precision what fraction of their ancestry derives from each of the major source populations. Modern British people are approximately 35 to 40% ancient hunter-gatherer in ancestry, with the remainder split between early Anatolian farmers and steppe pastoralists from the Pontic grasslands, in proportions that vary by region. Modern South Asian populations are weighted mixtures of ancient Iranian farmers and ancient Andamanese-related hunter-gatherers, with varying contributions from steppe ancestry depending on region and caste. Every modern population, examined with sufficient resolution, turns out to be a unique linear combination of ancient source populations, a specific weighted average of previously trained models, fine-tuned by thousands of years of subsequent selection in a specific environmental context. The genetic history of humanity is, in this sense, a sequence of ensemble models. Each generation, a new combination of previously developed solutions, tested by the environment and adjusted accordingly. One of the most consequential problems in modern AI research is called catastrophic forgetting. When a neural network is trained on a new task, the weight updates required to learn the new task tend to overwrite the weights that encoded the old task. The network gets better at the new problem and worse at the old one. The old knowledge is not stored somewhere safe while the new knowledge is being acquired. It is simply erased by the gradient updates that install the new knowledge in the same parameter space. Catastrophic forgetting is a fundamental obstacle to building AI systems that can learn continuously, that can acquire new knowledge throughout their operational lifetime without losing the knowledge they already have. Enormous research effort has gone into developing architectures and training strategies that mitigate this problem with varying degrees of success. The general principle that has emerged from this work is that resistance to catastrophic forgetting requires distributed, redundant representation. Knowledge that is not concentrated in specific weights, but spread across many of them simultaneously, so that updating any subset of weights does not eliminate the knowledge encoded in the rest. The human genome has solved this problem. Across a history of enormous change, continental dispersals, mass extinctions, agricultural revolutions, disease pandemics, the emergence and collapse of countless civilizations, the genetic record has maintained the signatures of events that occurred tens of thousands of years ago, visible and interpretable in the genomes of people alive today. The Toba bottleneck, the out-of-Africa migration, the Neanderthal admixture, the steppe expansion, all of these events occurred between 5,000 and 70,000 years ago, and all of them are detectable in modern genomes with precision and confidence. This is not trivial. In the intervening time, every one of the populations carrying these ancestral signatures has undergone additional bottlenecks, selection events, admixture pulses, and drift, all of which could, in principle, have erased the earlier signals. They did not. The signals persist because they are encoded in the distributed architecture of the genome, spread across thousands of loci simultaneously, each one contributing a small increment of information to the overall pattern, with no single locus carrying so much of the signal that its loss would eliminate it. This is the genomic equivalent of redundant distributed representation, the same property that machine learning researchers have identified as the key to resisting catastrophic forgetting. The practical consequence of this architectural property is that the human genome functions as a kind of geological record, a layered archive in which older events are visible beneath newer ones, each successive event writing on top of the previous one without fully erasing it. Ancient DNA research is essentially an act of stratigraphic reading, identifying and dating the successive layers of genetic information deposited by historical events, each one occupying a slightly different position in the frequency distribution of variants across the genome. There is a dimension of this that, when stated directly, carries an unexpected weight. Every person alive today is a living archive. Their genome contains, recoverable by anyone with the right tools, the traces of every population their ancestors belonged to, every migration their lineage participated in, every admixture event their ancestors survived, every bottleneck that shaped the line of descent leading to them. The information is encoded not in memory, not in language, not in any conscious record, but in the distributed structure of 3 billion base pairs of DNA maintained across every cell division for every generation, from the first humans to the present. You carry the memory of the Toba winter. You carry the memory of the first crossing into the Arabian Peninsula. You carry the trace of the encounter with Neanderthals. If your ancestry lies in Europe, you carry the genetic signature of 3 separate populations: ancient hunter-gatherers, Anatolian farmers, and steppe pastoralists, who met and merged in the Holocene to produce the modern European gene pool. If it lies in South Asia, you carry a different set of signatures, encoding a different history of meetings and mixtures. The network never forgets. It carries everything forward, compressed, distributed, layered, but intact. And the intelligence that evolved from it, 70,000 years after the journey began, is now learning to read it. John Holland was a mathematician and cognitive scientist at the University of Michigan, and in 1975 he published a book that would, in retrospect, mark one of the most important moments in the history of artificial intelligence. The book was called Adaptation in Natural and Artificial Systems, and its central argument was that natural selection, the process that had shaped every living thing on Earth, could serve as a template for solving hard computational optimization problems. If you wanted to find good solutions to a problem with a vast, complex, poorly understood solution space, you could do worse than to imitate what evolution did. Generate a population of candidate solutions, evaluate their performance, select the best performers to reproduce, combine elements of successful solutions to create new ones, and introduce occasional random mutations to explore the solution space. Then repeat for as many generations as it takes. Holland called his framework genetic algorithms, and the name has stuck. Genetic algorithms are now one of the oldest and most widely deployed optimization techniques in computational science, used in domains ranging from circuit design to drug discovery to logistics optimization to financial modeling. They are not the fastest optimization methods, and for problems with well-understood mathematical structure, there are usually more efficient approaches. But for problems whose solution space is vast, rugged, and opaque, problems where gradient descent fails because the landscape is full of discontinuities and the gradient is meaningless, genetic algorithms frequently find solutions that more elegant methods cannot. What Holland understood, and what his successors have continued to demonstrate, is that the effectiveness of evolutionary computation does not derive from any specific biological feature of natural selection. It derives from the abstract algorithmic properties: population-based search, selection pressure, recombination of successful partial solutions, mutation as exploration. These properties are not biological inventions. They are, in the language of computer science, a class of stochastic optimization algorithm, one that turns out to be particularly well suited to hard, high-dimensional, non-convex optimization problems. The fact that this class of algorithm was discovered by evolution 3 billion years ago and rediscovered by computer scientists 4 decades ago is not, by itself, surprising. Evolution, after all, is a good optimizer. Arguably the most powerful optimizer operating on Earth. The surprising finding is something subtler. When researchers use genetic algorithms and their descendants, evolutionary strategies, genetic programming, neuroevolution, to search for good neural network architectures, the architectures they discover are not arbitrary. They are consistent. They look like brains. Neuroevolution, the use of evolutionary search to discover the topology of neural networks rather than just their weights, is a relatively young subfield, but its results have been striking. When evolutionary algorithms are given the freedom to construct neural network architectures without any prior constraints on connectivity, they reliably converge on architectures with specific properties: sparse, scale-free connectivity, modular organization with clusters of densely connected nodes, hierarchical structure with general-purpose early layers and specific-purpose later ones. The same properties, again and again, regardless of the task, regardless of the specific evolutionary algorithm used, regardless of the initial population. These are, of course, the properties of the biological brain, but they are also the properties that Chapter 3 identified in the ancient human migration network, and the properties that Chapter 4 identified as characteristic of all well-trained deep neural networks: scale-free, hierarchical, robust. The evolutionary algorithm, searching freely through the space of possible network architectures, finds them every time. Not because it was instructed to, but because they represent the most efficient solution to the problem of information processing in a complex environment. The machines built in the image of evolution keep rediscovering the architecture of the systems that evolution built. The most striking result in the neuroevolution literature was published in a 2019 paper by researchers at DeepMind, working in collaboration with neuroscientists at University College London. The paper used a large-scale evolutionary search algorithm running on hundreds of computers in parallel over several weeks of computation to discover neural network architectures optimized for a suite of cognitive tasks, including spatial navigation, working memory, and sequence prediction. The evolutionary algorithm was given no information about the correct architecture. It was told only the tasks and the evaluation criterion. It started from a randomly generated population of architectures and evolved them over thousands of generations, applying selection, recombination, and mutation in the standard way. The architectures that emerged were not the standard convolutional or recurrent networks that human engineers had designed for these tasks. They were novel, sometimes counterintuitive, occasionally strange. But when the researchers examined them, they found something that they had not expected and that took some time to fully appreciate. The discovered architectures, mapped as graphs and analyzed for their structural properties, showed striking similarities to the connectivity structure of specific regions of the mammalian brain. The architectures best suited to spatial navigation shared topological properties with the hippocampal entorhinal system, the brain region responsible for spatial memory in mammals. The architectures best suited to working memory shared properties with prefrontal cortical networks. This was not a perfect one-to-one correspondence. The discovered architectures were simplified, abstract versions of the biological systems, missing much of the detailed structure that evolution had built into the real thing over millions of years. But the qualitative structural similarity, the degree to which unconstrained evolutionary search converged on architectures resembling specific brain regions when optimizing for the same functions those brain regions perform, was statistically significant and has since been replicated in multiple independent studies. The interpretation offered by the authors is careful and measured, as scientific interpretations should be. They suggest that the convergence reflects a deep connection between the computational demands of these cognitive tasks and the specific architectural solutions that can meet those demands efficiently. If the task demands certain computational properties, then any optimizer searching freely through the space of possible architectures, whether it is biological evolution operating over millions of years, or a computational evolutionary algorithm operating over weeks, will tend to find architectures that possess those properties. The architecture is shaped by the task, not by the substrate. This is convergent evolution at the computational level. Just as the physics of light demanded the lens and receptor architecture that both vertebrates and cephalopods independently developed, the computational demands of spatial navigation demand a specific class of network topology that both biological evolution and artificial evolutionary search independently discover. The demand is universal. The solution is universal. The substrates are different, but the architecture is the same. There is a further and even more recent finding that pushes this convergence into territory that becomes genuinely difficult to categorize. A 2022 study trained large language models on text describing human evolutionary history. Genome papers, population genetics literature, ancient DNA studies, and then analyzed the internal representations that these models developed for concepts related to migration, population structure, and genetic relatedness. The internal representations that emerged were not the representations that a human reading the same texts would develop. They were distributed numerical patterns spread across billions of weights, describing relationships in a high-dimensional space that has no human-scale intuition. But when these internal representations were visualized using dimensionality reduction techniques, the structures that emerged were recognizable. Population clusters separated by the same axes of variation that principal component analysis reveals in actual genetic data. Gradient structures corresponding to geographic clines. Cluster relationships reflecting the actual tree of human ancestral populations. A model trained on text about human genetics had, in the course of learning the language of population genetics, independently reconstructed the mathematical structure of the data that the language described. The language model had learned what the genome encodes, not by reading genomes, but by reading about them. The information was reconstructed from its description rather than from its direct observation, but the structure that emerged was the same structure that lives in the DNA of every person alive today. There is now a third direction of travel in the relationship between artificial intelligence and the human genome, not the use of evolutionary principles to build AI systems, not the discovery that AI systems converge on biological architectures. Something newer and, in a certain light, more vertiginous. The use of AI systems to decode the genome itself. Genomic deep learning. The application of neural networks to the analysis, interpretation, and prediction of genetic data has become one of the fastest growing subfields in both AI research and genomic science. The task is, in a certain sense, natural. The genome is a sequence, and sequence modeling is one of the domains in which deep learning has proven most powerful. Language models trained on protein sequences have learned representations of protein structure that rival the best physics-based models developed over decades. Convolutional networks trained on genomic sequences have learned to predict gene expression levels, chromatin accessibility, and the functional effects of genetic variants with accuracy that surpasses the prior state of the art by substantial margins. The most dramatic application of AI to genomic analysis, however, is in the domain of ancient DNA interpretation. Traditional analysis of ancient population structure requires making specific modeling assumptions about the number of source populations, the timing of admixture events, the demographic history of the lineages involved. These assumptions are always approximations, and they constrain what can be inferred from the data. Deep learning models trained on synthetic genetic data with known population histories have learned to infer those histories directly from the raw data, without requiring prior assumptions about the number or timing of events. Applied to real ancient DNA samples, these models have identified previously unknown admixture events in populations thought to be well characterized, and have dated known events with precision exceeding that of conventional methods. The human intelligence that evolved from the genomes being studied is now using tools built on evolutionary principles to reconstruct the history of those genomes. The decoder and the decoded are expressions of the same optimization process, separated by 70,000 years, and a vast difference in substrate, but structurally continuous. The brain that reads the genome is built from the genome. The tools the brain built to analyze the genome are built on principles derived from the genome's own evolutionary history. The circle, traced carefully, turns out to close. There is something important to notice about what this closing means and what it does not mean. It does not mean that the relationship between biology and computation is simple or fully understood. It does not mean that artificial intelligence is equivalent to biological intelligence, or that understanding the genome will solve the deep problems of consciousness and cognition. The circle closing is not an answer. It is a clarification of the question. The question, stated as precisely as possible, is this: is the mathematical structure shared by genetic evolution and artificial learning a coincidence of superficial description? Or is it evidence that both systems are governed by the same underlying principles? Principles that apply to any sufficiently complex information processing system operating under adaptive pressure? The evidence reviewed in this chapter, and in the chapters before it, suggests that it is the latter. Not with certainty. Science rarely provides certainty, but with a convergence of independent lines of evidence that makes the coincidence, hypothesis, increasingly difficult to maintain. And if it is the latter, if the mathematics of adaptive information processing is genuinely universal, as the physics of optics and the chemistry of energy metabolism are universal, then the implications extend well beyond the specific domains of genetics and machine learning. They reach into every field that concerns itself with how minds arise from matter, how knowledge accumulates in physical systems, and what it means for a system to understand itself. Chapter 10 attempts to draw those implications out carefully without overreaching. But first, it is worth sitting quietly with the observation that brought us here. Two maps, identical in structure, separated by 70,000 years, built from entirely different materials, for entirely different reasons, by entirely different processes. Asking the same question, finding the same answer. Pull back now, far back. Look at everything that has been covered, not as a sequence of arguments, but as a single assembled picture, and ask what it shows. It shows two systems, one biological, one computational, one unfolding across geological time, the other built in decades. One encoded in the chemistry of living cells, the other in the physics of semiconductor circuits. Both, when examined closely enough, performing the same operation, taking in information about an uncertain environment, adjusting an internal representation in response to the gap between prediction and reality, and producing outputs that are progressively better calibrated to the actual structure of that environment. At the topological level, both systems are scale-free networks. The degree distribution of connections, the pattern describing how many connections each node has, follows a power law in both cases. Most nodes have few connections. A small number have very many. The ratio between these two populations follows the same mathematical function. This has been measured. It is not an impression. It is a number that falls in the same range whether the system being measured is the global pattern of ancient human migration or the weight matrix of a trained deep neural network. At the mechanistic level, both systems use structurally equivalent processes to learn and adapt. Population bottlenecks and dropout regularization both enforce compression, both prevent overspecialization, both produce systems that generalize more effectively than their uncompressed predecessors. Gene flow and backpropagation both transmit corrective information from one part of the system to another. Both adjust the global architecture in response to local feedback. Both produce modifications that propagate through the entire system rather than remaining confined to the point of entry. Founder effects and weight initialization both establish starting conditions that cast shadows over the entire subsequent trajectory of the system, biasing the space of accessible outcomes without determining any single one. At the mathematical level, both systems are instances of stochastic gradient descent on a high-dimensional optimization surface. Natural selection is gradient descent on the fitness landscape. Backpropagation is gradient descent on the loss surface. Both use noise, genetic drift in one case, stochastic batch sampling in the other. To escape local minima, both navigate rugged high-dimensional surfaces toward regions of low loss. Both can be formally described with the same class of stochastic optimization equations, with the biological variables translated into their computational equivalents and the computational variables translated back. 3 levels: topology, mechanism, mathematics, each one independently pointing in the same direction. These systems are not merely analogous. They are structurally equivalent. They are 2 instances of the same class of adaptive information processing network operating across different substrates and timescales, but governed by the same fundamental principles. The theoretical framework that best explains this equivalence is one that has been developing at the intersection of information theory, statistical physics, and machine learning for the past 2 decades. It holds that any sufficiently complex system capable of representing and transforming information will, if subjected to optimization pressure over sufficient time, converge on a specific and universal class of architectures. Architectures defined by scale-free connectivity. hierarchical feature extraction, and robustness through redundancy. This convergence is not the result of any specific biological or computational mechanism. It is the result of the mathematical constraints imposed by the optimization problem itself. The problem is, how do you move useful information reliably through a noisy, unpredictable environment? The answer, derived independently by biological evolution and artificial intelligence research, is, with a scale-free, hierarchical, redundant network that can compress its representations under pressure and propagate corrective information across its full extent. This architecture is not the only possible one. It is not even necessarily the optimal one in all cases, but it is the one that consistently emerges from optimization processes operating on the class of problems that both living systems and learning machines face. The universe has a preferred architecture for moving information. Both life and computation have been finding it, convergently, independently, and completely. The findings described in this documentary are, in the strict scientific sense, findings about mathematical structure. They concern the topology of networks, the dynamics of optimization algorithms, the statistical properties of data distributions. They do not, by themselves, resolve any of the deep philosophical questions about consciousness, meaning, or human uniqueness. They do not tell us whether machines can think, or whether thinking is computation, or whether the universe is in some sense alive. But they do change the shape of those questions in ways that may be more important than any specific answer would be. For the science of human origins, the convergence described in this documentary suggests that the mathematical tools developed for machine learning are not external impositions on the genetic data, not translations of a biological subject into a foreign computational language. They are natural descriptions, formalisms that accurately capture the structure of the genetic data, because that structure is, at a deep level, the same kind of structure that machine learning systems also instantiate. This means that the entire toolkit of modern AI, the dimensionality reduction methods, the optimization theory, the information theoretic frameworks, is directly applicable to the study of human evolutionary history, not as an approximation or an analogy, but as a precise formal language for describing what actually happened. For the science of artificial intelligence, the convergence suggests that the evolutionary record is not merely historical context, but a design resource. 3 billion years of optimization on the hardest possible problems, survival and reproduction in a changing world, have produced a catalogue of architectural solutions encoded in living genomes whose formal properties are now mathematically legible. The brain's scale-free, hierarchical, robust connectivity is not a biological curiosity. It is an engineering specification derived from first principles by the most extensive optimization process ever observed. Understanding it in formal terms, which the convergence between genetic and computational science is beginning to make possible, may be the most productive path toward building AI systems that are genuinely robust, genuinely generalizable, and genuinely capable of continuous learning without catastrophic forgetting. For the philosophy of mind, the convergence raises a question that is harder to frame, but may be the most important of the three. If two systems, as different as a biological population and a silicon neural network, can be formally equivalent, not metaphorically similar, but mathematically identical in their structural and dynamic properties, then the substrate distinction that underlies most of our intuitions about the difference between life and machine, between biology and technology, may be less fundamental than it appears. Not because machines are becoming biological, not because life is mechanical, but because both are instantiations of mathematical structures that are substrate independent, that arise wherever and whenever the optimization problem they solve is posed. You are a node in the human network. Your genome is a trained model, shaped by 70,000 years of gradient descent on a fitness landscape that you have never seen and could not survive alone. The intelligence you use to think about this, the brain that processes these words, finds them interesting, holds them in working memory, connects them to other ideas, was built by the same optimization process, running on the same principles, producing the same scale-free, hierarchical, robust architecture that machine learning engineers keep independently rediscovering. The boundary between what you are and what the machines are is not drawn where most people assume it is drawn. It runs through much stranger territory, through the mathematics of optimization, through the information theory of heredity, through the network topology of learning systems that share, across an almost incomprehensible gulf of time and substrate, the same deep structural solution to the same deep problem. You did not begin as information, but you have always been information, encoded and transmitted and transformed for as long as life has existed. And the machines built to process information are, in ways that are still being understood, built in your image, not intentionally, but inevitably, because the problem they solve is the same problem your ancestors faced on the shore of Africa 70,000 years ago, with the whole empty world in front of them. Return to the beginning. Two maps. One assembled from genetic data collected across decades, representing the movement of human populations across 200,000 years and every continent on Earth. One rendered in milliseconds, representing the internal state of a neural network that was trained last week on data that has nothing to do with human migration, ancient history, or evolutionary biology. The shapes are the same. After everything explored in this documentary, the genetic timestamps, the haplogroup trees, the bottleneck events, the gradient descent on fitness landscapes, the transfer learning of admixture, the convergent architecture of neuroevolution, the shapes are still the same. The explanation for why they are the same has been elaborated considerably, but the central observation has not changed. it is still, at its core, a mystery. Not a mystery in the sense of something unknown. The mathematical structure of the correspondence is known in some detail, and the theoretical framework that accounts for it is increasingly well developed. A mystery in a deeper sense, the sense of something whose full significance cannot yet be calculated. The convergence of genetic and computational intelligence is known. What it means for the future of both fields, for the philosophy of mind, for the long-term development of artificial systems, is still being worked out. One of the oldest and most repeated findings in the history of science is that nature does not waste solutions. When a particular mathematical structure proves to be effective, when it consistently solves a problem better than competing structures, it appears again and again in different contexts, in different substrates, assembled independently by different processes. The helix as a stable structure for carrying information in a linear polymer. The sphere as the shape that minimizes surface area for a given volume. The power law distribution in network after network after network. The solutions are discovered by physics, by chemistry, by biology, by mathematics, and they turn out to be the same solutions. The scale-free, hierarchical, robust information processing network is one of these solutions. It appears in the neural connectivity of brains. It appears in the trained weights of artificial neural networks. It appears in the migration record of the human species. It appears in the architecture of the internet. It appears in varying forms, in gene regulatory networks, in protein interaction maps, in the citation networks of scientific literature, in the spread of language through populations. Wherever you have a system that must move information across a complex environment and adapt to feedback about how well it is doing, you tend to find, eventually, some version of this architecture. The universe is not indifferent to how information organizes itself. It has preferences expressed in the mathematics of optimization and the physics of networks that consistently favor certain structures over others. Life discovered these structures 3 billion years ago. Human intelligence discovered them 70,000 years ago in the form of social and cognitive architectures for coordinating behavior across groups. Artificial intelligence discovered them in the past half-century in the form of trained neural networks that keep converging on biological architectures regardless of how they are initialized. There is a single genetic marker introduced early in the story that sits in the genome of every person alive today whose ancestry lies outside Africa. It is not functional. It does not make you better or worse at anything measurable. It is simply a record, a transcription error made in a single individual somewhere in East Africa, roughly 70,000 years ago, that happened to be present in the small group that crossed into the wider world and became the ancestors of everyone who followed. Somewhere in a server farm, a neural network is running. It is processing data at a speed that would have been incomprehensible to those ancestral humans. It does not know what it is. It has no concept of its own history. It is doing what it does, minimizing a loss function, following a gradient, searching a landscape it cannot see, guided only by the incremental signal of error and correction. It does not know that it is moving the same way those humans moved. It does not know that its architecture echoes theirs. It does not know that the mathematics governing its search is the same mathematics that carried the first members of your lineage out of Africa and into the whole uncertain world. But you do, and perhaps that is the beginning of something.